These are the recorded lectures for Chapter 9 on the cell cycle and cellular reproduction. Primarily, what this chapter is going to cover is actually the process of mitosis. So we have Chapter 9 that covers mitosis, and then Chapter 10 that covers meiosis. There are 5 sections. We will focus primarily on the first 4, and we will discuss how mitosis is related to cancer. Okay, so the definition of the cell cycle, excuse me, it is an orderly set of stages from the first division of a eukaryotic cell to the time the resulting daughter cells divide. So basically from the time that a cell, the cell's life begins until it divides into 2 new cells. Now, before the cell divides before a cell divides again, it's going to grow larger. It's going to double its organelles because each new cell will need a copy, and it's going to replicate or copy its DNA because, again, each new daughter cell is going to need a copy of the DNA from the parent cell. So these are things that are going to happen during the process before the next cell division. There are 2 major stages of the cell cycle. There's interphase, which includes 3 stages, G1, S, and G2, and really it includes 4 if you count G0, which is the stage that cells go into when they aren't going to divide again. So interphase has 3 stages. The mitotic stage, or the M stage, is the actual cell division part, and it includes mitosis and cytokinesis. And this is a pie graph showing you the stages of the cell cycle. It also shows these little stop sign symbols show the checkpoints, which are places during the cell cycle that are basically for determining whether the cell will go on to the next phase, because sometimes there's a problem and the cell really shouldn't divide. So this pie graph also lets you know basically for most cells that are actively dividing, the relative amount of time that the cell spends in each phase. So obviously what's highlighted here in purple, interphase, the cell spends the majority of its time in interphase and then less of its time actively dividing in the M stage, mitosis and cytokinesis. So if we start with interphase, the G1 stage is the growth stage. And this is the stage where the organelles are going to be replicated. They're going to be doubled. So organelles like mitochondria, lysosomes, Golgi, endoplasmic reticulum, those organelles are going to copy themselves so that there will be there will be 2 of each one of them. There is a checkpoint called the G1 checkpoint. This is the cell cycle main checkpoint. If the DNA is damaged at this checkpoint, apoptosis will occur, and apoptosis is programmed cell death. If the DNA is not damaged, the cell is committed to divide when growth signals are present and nutrients are available. So if the DNA is not damaged at the G1 checkpoint, then it will move on, the cell will move on to the S stage of interphase. And this S stands for synthesis. So this is when the cell will grow more and DNA will replicate. This is the DNA replication stage of interphase. After the S stage, the cell will enter G2, which is a second growth stage. And during this stage, the cell is going to produce the proteins needed for cell division, like spindle fibers, which are microtubules. there is a checkpoint called the G2 checkpoint, and this is the checkpoint for mitosis. This is to, if the cell passes this checkpoint, it will enter mitosis. So it says mitosis will occur if DNA has replicated properly. Apoptosis will occur if the DNA is damaged and cannot be repaired. So as long as the cell is ready after the G2 checkpoint to enter mitosis, then, or the M stage, it will undergo mitosis, which consists of prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Cytokinesis is the part where the cell actually splits in half, that divides in half. After metaphase, we call this the M checkpoint. This is the checkpoint to see that the spindle is assembled properly. This is the spindle fibers which attach to the chromosomes and help to organize them and then pull them apart so that each daughter cell gets a copy of each chromosome. So at this checkpoint, mitosis will not continue if the chromosomes are not properly aligned. So we're looking for the chromosomes to be lined up at what we call the metaphase plate or the equator of the cell. Now, everything I just said is in the notes, so we'll go through it again, but now we'll look at the notes. So an overview of interphase. Most of the cell cycle is spent in interphase, which we saw that from the graph. But what I highlighted here is that nerve and muscle cells do not complete the cell cycle because nerve cells and muscle cells don't keep dividing once they're produced. They remain in the G0 stage. So basically, they remain in interphase for the remainder of their life. Now, this slide, slide 6, goes through the stages of interphase, which are in an actively dividing cell. They are G1, S, and G2. Interphase takes up 90% of the cell cycle. If you go back to the pie graph, this is a little off, isn't it? This looks like this looks like interphase takes up 75%, but it's a little bit off because whoever illustrated the graph is trying to get in the names of all the stages of the M stage. So anyway, but it's actually 90%, so that the amount of time that the cell spends in the M stage, mitosis and cytokinesis, is more like 10%. Okay, so the G1 stage, the primary things that happen during that stage is that the cell doubles its organelles and grows in size. The S stage, or synthesis, is when DNA replication takes place. The G2 stage is when the cell synthesizes proteins, including microtubules, which are going to be part of the spindle assembly. So these proteins are synthesized during the G2 stage, and they are necessary for the cell to divide. So that you can understand what happens during the S stage. Before the S stage, each cell has a, depending on the number of chromosomes for that cell, so let's just say it's a human cell. Each human cell has 46 of these single-stranded chromosomes consisting of one chromatid. After DNA replication, this is still a chromosome. This one here that looks like an X, it's still considered one chromosome, but it is a duplicated chromosome. So it's going to contain two sister chromatids that are joined together at a region called the centromere. So the entire structure is called a chromosome. It does contain two sister chromatids that are joined together at a region called the centromere. And then after the cell divides, again, the daughter cells are going to have these single-stranded chromosomes that have only one chromatid until they undergo their own S phase. Now, the M or mitotic stage includes mitosis and cytokinesis. Mitosis is the division of the nucleus. This is the part where the chromosomes that are located inside the nucleus will separate because they consist of two sister chromatids. So those two sister chromatids will separate. One will go into one daughter cell and the other will go into the other daughter cell. So it's the division of the nucleus, or nuclear division, and the daughter chromosomes are distributed by the mitotic spindle, the spindle assembly, which consists of the microtubules, also known as spindle fibers. And those daughter chromosomes will be distributed to the two daughter nuclei in the two daughter cells. All right, then we have cytokinesis, which occurs at the end of mitosis. They occur simultaneously, but it starts during the last stage of mitosis. And cytokinesis is division of the cytoplasm. And the M stage results in two genetically identical daughter cells. So if we had a human cell that the parent cell has 46 chromosomes, the two daughter cells are going to also have 46 chromosomes. And they will be genetically identical. Clones, in other words. Mitosis is a process of cloning cells. Okay? Now, the cell cycle does have, there are ways to control the cell cycle. Okay, so to control whether or not it proceeds, whether or not the cell divides, based on whether or not the cell is healthy. So a signal is an agent that influences the activities of a cell. And we have two types of signals that we'll talk about. There are growth factors and cyclins. Growth factors are signaling proteins received at the plasma membrane. So they come from outside the cell. Then cyclins are inside the cell, and they increase and decrease as the cell cycle continues. So without cyclins, the cell cycle will stop at the G1M or G2 checkpoints. So the presence of these cyclins is important at each of the checkpoints during the cell cycle. And if they're not present in sufficient quantities, the cell cycle will stop at those checkpoints. Apoptosis, we've learned this before, but apoptosis is programmed cell death. It's caused by enzymes called caspases. Cell division and apoptosis are opposing forces. What cell division does is increase cell numbers. It helps cells to multiply. Apoptosis decreases cell numbers. And this is an image of illustration of what happens during apoptosis. If these are normal cells here on the left, if the cell is going to undergo programmed cell death or apoptosis, it will first become rounded, kind of blow up, and the nucleus will begin to collapse. It forms these bulges in it. Then you will see the nucleus breaking down further. You will see the plasma membrane forming these blisters and blebs, is what they're called. And then finally the cell's going to start to fragment. An abnormal cell that could become cancerous or a cell infected with a virus will die through apoptosis normally. One of the signaling proteins that's important is called P53, and we'll look at P53 later when we talk about cancer. P53 is a protein that stops the cell cycle at G1 when the cell's DNA is damaged, and it initiates an attempt at DNA repair. So if the DNA can be repaired, the cell will continue on to the S stage of interphase. But if not, then it won't. All right, second part of chapter 9 is on the eukaryotic chromosome. So in eukaryotic cells, the chromosome consists of DNA, a single DNA molecule, which is shaped like a double helix or a twisted ladder shape. In other words, the DNA molecule is double-stranded. the DNA to get